Next, it's a great pleasure for me to introduce Dr. Becky Crean. She is the Executive Director of Clinical Development at Ionis Pharmaceuticals. Becky has been really incredible, actually, through the ABOM initiative, and she is one of the few people on the we have a steering committee of people that really take the lead and participate and push to make a difference and spend the time and volunteer their time to help the ABOM program, and Becky's been definitely one of them. Her and Rob have just been incredible in their support of ABOM, so I want to certainly thank Becky for all of her help and all of her insight. So Becky, if you want to come up. She's going to be speaking to us today on an update on the HALOS clinical trial for individuals with Angelman syndrome, which is an antisense oligonucleotide as well. Thank you.
Thanks, Allison. Thanks, everyone, for allowing me to be here today. Thank you to FAST for inviting us. I think I speak on behalf of all of us at Ionis and also our partners at Biogen that we really are honored to be here and to be sharing our updates with our program with you today. We've been involved in the community for quite a few years now and have been coming to the meeting, and it really is remarkable to see how much progress has been made year after year, even in the midst of COVID on top of it. So it really is exciting for all of us. It's really unusual in a field, in a disease like Angelman syndrome, a rare disease, to have so many treatments pushing towards the clinic, in the clinic at the same time. You've got multiple drugs in the clinic right now testing for safety and efficacy, all with different mechanisms of action. You've got ASOs, each with their own chemistry. You've got gene therapy, small molecules, all in or near the clinic. And so it's quite energizing for all of us back home at Ionis. We have a lot of scientists and dedicated people on this Angelman team, and it really does provide a lot of inspiration and energy to help us do what we do each day. So I want to thank you for that. So as you know, Ionis has been in the ASO business for quite some time. We really pioneered this development of turning antisense oligonucleotides into a therapeutic. We're a bit unique in the biotech industry in that our sole focus is on developing ASOs. And 100% of our people back in the offices and labs are focused on that. It would seem a little bit stubborn to some, I would say. We've been at this for about 30 years now, and we've had some successes and some setbacks along the way, but we've stuck with it. Our whole group back at Ionis is focused on this endeavor. A lot of people said it wouldn't work. Some companies tried it, it didn't work, they got out of it, but in a sort of, as I mentioned, stubborn way, we stuck with it. And we think we've brought some really important medicines to people who need them with various disorders and medical conditions. So we're doing the same with Angelman. And we've embarked on this ASO development for Angelman syndrome with a compound that we call Ion 582. It's an ASO, but not all ASOs are developed the same. You'll hear a lot of different types of ASOs that get developed, and you'll hear things like MOs, LNAs, O-methyls, a lot of different types. This one in particular, Ion 582, is an ASO that is designed to stop the silencing mechanism on the paternal UBE3A gene. And as we've heard, how that'll work is that it then is thought to produce the UBE3A protein in the brain. This particular ASO happens to be what we call a 2' Mo antisense oligonucleotide. And if you look kind of at the bottom left of the screen, it's a chemical structure. I won't ask you to look too hard, but it just shows what this particular type of ASO is. We call it a gapmer, which is designed to activate the RNase H cleaving mechanism. So you've got the Mo modifications are at the ends. You've got the DNA in the middle, and you've got the phosphorothioate throughout the molecule. And By that type of chemical modification, like I said, it activates this RNase H cleaving mechanism, which is targeted to a specific strand on the antisense to cut that off and make sure that it doesn't stop the UBE3A expression in the CNS. So not all ASOs are the same. As I mentioned, you've got LNAs, you've got O-methyls, and they're all designed for different reasons, and you choose different chemical modifications based on what you're trying to achieve with the compound. And so the MO compounds, the MO chemical modifications are important to us, and ones that we have found that when you're trying to deliver into the brain, they tend to be selected for greater potency at reducing the target RNA. They have increased tolerability. Their duration of action is quite long. And so those are the ones we focus on at ASO for disorders of where we want to put the drug into the brain. And that's true for this compound ION 582 that we're developing for Angelman syndrome. So it's a long process. We start out in choosing these ASOs based on a really wide funnel. We start with testing over 3,000 ASOs, specific MO chemical modifications, that first we select them for the ones that bind to that target of interest. So in this case, it's the antisense RNA on the UBE3A gene. So we go through a huge screening process for that, and from there, a handful drop down into the bucket that look good and that have the properties that we want to see in this target reduction. So from there, what we do is we take the remaining ASOs and we start to test them in cells and animal lines. We look at transgenic mice, and that's where you have the mice that are engineered with the human gene. We look at Angelman syndrome mouse models. We look at iPSC neurons, and each step along the way, we're testing all of these different ASOs to see how effective they are, how well they're tolerated, how well they are at knocking down the thing that we want it to knock down, and if the protein is expressed the way we want it to be. So it's a kind of a constant process of choosing the right molecule to take to the next level. And as I mentioned, we take sort of a broad approach to this and start to narrow it down after each of these tests. So then we're left with a handful of them, and we take those into the larger animals. So the ones that look promising across all the board, we take those into non-human primate studies. We look in toxicology studies. Those are really sort of the IND-enabling studies that you would do in order to select the one that appears to be safest, most well-tolerated, and most efficacious into humans. So ION 582 went through this entire process. It started out in the big bucket up top and found its way down to the bottom, and we chose that one because of its properties, the properties that we talk about in terms of potency, duration of action, how well tolerated they are. So for the test that we do in all of these studies, we look at doses and dosing regimens that far exceed what we would expect to do in the clinic, because we want to see the whole range of what the compound will do. So for this particular compound, we've shown that in all of these animal models that have been shown to be safe, well tolerated, and also at doses and dose intervals that are much higher than what we're doing in the clinic. So it gives us a lot of confidence that we have a wide range to look at in the clinic, and that what we do look at in the clinic is thought to be safe and well tolerated in the early stages of the program. So we are confident that all individuals with Angelman syndrome may benefit from this particular compound. We're starting with the HALOS trial, and in that trial it's small, but it does include quite a broad range of ages. We're starting with toddlers age 2 years and up through adulthood. We are focusing on deletion and mutation genotypes for this first-in-human study. We do have to take a pretty methodical approach to this in the very beginning. As you just heard Emil say, You only know so much from the animals, and then you learn more once you get into the clinic. So you start small, you take a thoughtful, sort of measured, systematic approach, and each step along the way you gain more and more information about that particular compound and how it behaves in human beings, and then you can build upon each of those studies. So that's what we're doing in this first trial. We do anticipate being able to broaden this to other ages and genotypes. We know that from the HALOS trial, we'll start to get information about what the compound will do in the broad age range, but also only with mutations that have one copy of the paternal allele. So we'll know what this compound can do when you want to turn on one copy. It won't tell us everything. It won't tell us how this compound might work in newborns and really young babies. We're going down to 2, which is pretty young, but it's a whole different ballgame when you get to newborns and infants. And also the genotypes that have potentially 2 copies of the paternal allele to turn on. We don't know what it means to potentially double normal amounts of UBE3A in the body. We've heard a little bit about the science on that and the progress towards that. I think there's a lot more we need to know, but we'll also gain information from the current HALOS trial, and we'll use that information to build on our knowledge so that we can plan for the next steps. And all of this is to say that we very much think that this compound will work. I think I agree with Emil, is that there's not a huge concern. We just have to do the right studies and get the right information before we broaden it to other populations and age groups. So the HALOS study is a really important trial. It's our first in human, as all of them are here today. It is designed really to answer a very specific question, and that question is it safe, and can people tolerate the compound in the clinic? And as you go through the phases, we've talked about this before, each trial and each phase has a different question to answer. So as I mentioned, this is the first one. Our primary focus is safety, and we want to look at different dose levels and dose intervals of the compound and see how people respond to these different regimens. That's important knowledge that we need, and we have to have it first before we can then make decisions about what to do next with it. So we started the trial earlier this year, about a year ago. The trial is active and still ongoing. As I mentioned, this is an ASO. As all ASOs, this one's administered intrathecally into the spine with a lumbar puncture. The trial is in individuals with Angelman syndrome, and we are giving different types of sedation and anesthesia to help kids tolerate the intrathecal injection. So the study design, it is a phase 1/2. As I mentioned, it's our first trial in people. We are giving several doses of the drug, dose levels, and also at different dose intervals over about a 3-month period. Everybody receives active drug. There is no placebo in this, and you just heard Emil talk about why some of the reasons are for that in these different phases. So this one is open label. In terms of who participates, as I mentioned, it is a small trial. These first ones usually are. Roughly about 45 individuals, males and females with Angelman syndrome, broad age range, 2 to 50 years old, but where they come into the program and at what point, we are staggering different age groups. So depending on your age will determine when you're eligible to come into the trial. And again, we are looking at those with deletion and mutation genotypes in this early trial. So during a family's participation, both the individual living with Angelman syndrome and their family take a lot of tests. They answer a lot of questions. We look at a lot of different types of functioning, things that are important to families and that you want to see a treatment improve, things like language, motor skills, learning new information, seizures, sleep. We'll be looking at all of that in this early trial. Some of that's done in the clinic each time you come into the clinic for a visit, but sometimes it's done at home as well. We want to better understand what happens in the home in sort of your natural environment. So we think those are important to start including early on as well. And as I mentioned, the goal in this first study really is about the safety and tolerability of this compound. That's our primary goal, and that's what this study is specifically designed to answer and to measure. But we are measuring all areas of function, as I mentioned. We do want to start to get an early sense of what the effects might be. You've seen in some other studies that have presented that they're starting to see effects, and we would expect that in this one as well. The other purpose of this trial, though, is really to generate the data that we need to start looking at optimal doses and optimal dose regimens for the next phase. This is just the first one. There's going to be more trials as we gain new information about this compound, and we'll start, it's kind of like that funnel I showed with the ASOs. You start to get more information and you start to hone in on the right dose levels and right dosing regimen with this compound, and that's what we're doing with this trial. So it is a small trial, but we do have a pretty big global reach with it. We've got, I think, 13 or 14 sites spread out across 9 countries, which is great. But what it also means is that because it is such a small trial with so many countries involved and sites, it means each country will probably only enroll a few families at each of their sites. But we also think that it's important that we start to understand how the drug behaves across the globe, different geographical regions and different populations. So that's why we've broadened our reach so big in this first trial, even though it's very small. I mean, I think you've heard from some of the sites here. One site could probably enroll the entire trial, but to really understand how the compound works, we think it's important to spread out a little bit even in these early trials. So how are we doing so far? Well, I will just, before I say how we're doing, I will just acknowledge that drug discovery is an incredibly long process. It can take anywhere from 15 to 20 years to start on a project in the lab, start to develop a drug and get it all the way through the discovery process, into the clinic, the development process, through all the phases, and actually be made available for the patients who need them. 20 years, that's a long time. We've also heard that in rare disease, we have a little bit more flexibility, I think, to combine some of our studies and get to our answers quicker for good reason. We're able to do that here as well, but we do acknowledge that it's a long process, but it's an important one to be thoughtful and thorough with. So it does take time, but there's a reason for that, and we're trying to be thoughtful and purposeful in these early studies as well, so that we can get the information we need in the quickest way possible. Now, having said that, we know that we wouldn't be here today without families like yourself who are interested and engaged and want to be involved in this process, and it's because of that and your interest that we've been able to make progress in this program, and we're actually right on time and right where we think we need to be with this program. And we thank you for that, and we know you do this also knowing that there's no guarantee that there would be any benefit for you or your family by participating in any of these research trials, whether it's natural history or these clinical trials, especially these earlier ones. So we thank you for that, and because of that, we've made good progress this last year. So this trial, as I mentioned, we're looking at different dose levels, and so how it works is we look at a dose level in a small group of individuals, and we give several doses and assess them over a period of time. We look at the safety and some of the clinical measures, and so while that's happening, we don't dose other people. So we take this group, they dose at the level, we evaluate it, and then once we have the data we need from that particular dose level and regimen, then we can go into the next one, and we can go to a higher dose or spread out the doses or whatever the design might be. So that's what we're doing. We're taking a very systematic approach so that we can get the kind of data we need to make an educated decision about the next phase. So all of that's going well. We've been able to look at multiple dose levels of this compound in the study so far. And while I won't give specific information about data in an ongoing trial, what I can tell you is that all the doses that we've tested so far in this study have been well tolerated. We haven't had any drug-related adverse events or safety signals that have been identified. But again, we won't go into details about the compound while the trial is active, but just to let you know that things are going well and as planned. So the other thing we've been able to do this past year is we've been able to launch our Part 2 long-term extension for the trial. This will be a continuation of the current trial. And what will happen is after families go through the first 3 months with dosing, then they'll get to roll into what we're calling Part 2, this long-term extension, and continue on treatment for an additional 12 months. So you've got the Part 1, which is the part I've just talked about, assessing the safety and tolerability of different dose levels and intervals of the compound over a really short period of time, like 3 months. So you can get good information out of that, but it doesn't tell you what happens when you dose longer with the compound. So we've been able to select doses and intervals from this first part of the trial in order to carry forward into longer-term dosing, which supports the Part 2 long-term extension. So again, our primary focus will still be safety and tolerability with this long-term dosing. We'll also be able to start to look at the best dose and best interval and how it might impact clinical benefit for different ages and different genotypes in this study. So now we're really starting to peel back the layers and see how it might be acting differently depending on your age and your genotype. So this long-term extension, additional 12 months of dosing, will allow us to start to understand the compound better in that context. So where do we go from here? As I mentioned, we've made great progress this year. There's a lot more to do. Our goal really over the next year, as we complete parts 1 and 2 of the HALOS trial, is to better understand the safety and tolerability, start to get an idea of any clinical changes that might be taking place in the trial. And this will allow us then to take that information and start to think about the next phase of the program. I mentioned earlier that we do think that this compound will work in all ages and all genotypes, so we definitely are thinking about expansion to newborns, additional genotypes, but that will be driven by the data generated from the HALOS trial. So it's really important that we complete this first trial, look at the data, see what it tells us, and how that might drive the next phase in expanding to different populations. Another important thing we'll do over the next year is we'll continue to analyze the CSF, UBE3A in our industry-sponsored natural history studies. You've heard a little bit about that. These are really important studies because as we think about expanding into younger ages and different genotypes, we want to understand what the levels of UBE3A are in the CSF, not only just at a single time point, like whether it's there or not and how much is there, but also how that might change over time, just natural variation. So an important part of this work and completing this work is looking at those longitudinal samples, and we have an ongoing study that's doing that as well. That'll help us really better understand that if we see changes in one of our treatment trials, we can better understand if those changes in levels are due to just natural variation or different ages or different genotypes, and which part is contributed to the differences in the drug as well. So those are important studies to finish. And all this to say that as we gain more of this information that we've gained last year and what we hope to do next year, that will really help us determine the safest and most efficacious dose from the HALOS trial that we can then test in the next phase of development. So we're actively doing that day in and day out. And that's our goal for this next year. So with that, I just want to say thank you to the entire Angelman community. As I mentioned before, we wouldn't have made this kind of progress without your involvement and contributions. Thank you to FAST for having us and all the Angelman organizations around the globe who are doing really important work. And I know they couldn't do it without you either, so thank you.